The moment a single drop of your blood hits the glass, it holds the complete chemical signature of your blood type. Today, we're going to decode that signature in real time using a simple diagnostic test. Human red blood cells carry over 400 different surface antigens. But when it comes to clinical medicine, two specific classification systems demand our immediate attention: the ABO system and the RH system. If a patient receives the wrong blood type during a transfusion, their body's immune system recognizes the donor cells as foreign invaders. The recipient's antibodies will immediately bind to and destroy those introduced red blood cells. This destructive reaction can be fatal. Accurately matching the donor and recipient blood types is the strict, non-negotiable requirement for preventing severe transfusion reactions. This diagram shows the relationship between antigens and antibodies. Think of antigens as distinct shape markers attached to the surface of your red blood cells, while antibodies are the Y-shaped defenders floating in your blood plasma. Look closely at the bottom section. When an antibody encounters its exact matching antigen, they physically lock together. This causes the red blood cells to clump up into tight, visible masses, a biological reaction known as hemagglutination. Laboratories use two distinct methods to map these interactions. Reverse typing tests the patient's plasma against known lab cells. Forward typing tests the patient's actual red blood cells against known lab-grade antibodies. Today, we will perform a forward typing test. By deliberately introducing specific antibodies to a blood sample, we can trigger that visible hemagglutination process and reveal exactly which surface antigens are hiding on your cells. Step 1 is preparing the materials. Remove your ABO blood grouping kit from the refrigerator. Gently invert each vial 3 to 5 times to ensure the contents are perfectly mixed. Then let them sit until they reach room temperature. Here we have the 3 standardized antibodies used for testing, and their color codes are universal. The blue liquid is anti-A, the yellow is anti-B, and the colorless or gray-capped vial is anti-D, which tests for the RH factor. Next, place a clean glass slide on a white sheet of paper or a porcelain tile. The white background will make the visual reaction easier to identify later. Mark 3 distinct sections on the slide: A, B, and RH. Getting the reagents to the proper temperature and clearly labeling your workspace ensures you don't make any basic procedural errors before the blood is even drawn. Now it's time to collect the sample. Take an alcohol swab and thoroughly surface sterilize the tip of the ring finger on your non-dominant hand. Wait a few moments to allow the alcohol to completely air dry. Using a brand new, sterile lancet, give the fingertip a brisk, gentle prick. As the blood starts to pool, take a sterile cotton ball and wipe away the very first drop that oozes out to ensure a clean sample. Apply gentle pressure to the finger. Allow one distinct drop of blood to fall directly into each of the 3 labeled sections on your glass slide. Keep them spaced out evenly. Placing these sterile, perfectly isolated drops of blood creates an uncontaminated baseline, giving us 3 separate identical canvases for our chemical tests. Now we introduce the antibodies. Add exactly 1 drop of the blue anti-A reagent to the first blood drop. Add 1 drop of the yellow anti-B to the second drop, and 1 drop of the colorless anti-D to the third drop. When doing this, hold the vials at a slight height above the slide. You must take absolute care to never let the dropper tips touch the blood samples, which would instantly ruin the reagents in the bottle. Once the reagents are dispensed, it is time to mix. You must use a separate, brand new toothpick for each individual drop. If you cross-contaminate the mixtures by using the same stick, you will trigger false positive results and invalidate the entire test. Thoroughly mix each drop in small circles to combine the blood and the reagent. Leave the slide undisturbed for about 30 seconds. After the time is up, pick up the slide and gently rock it back and forth to encourage the antigen-antibody reactions. By perfectly isolating both our chemical reagents and our mixing tools, we can be confident that any visible reaction we see is a genuine biological response, not a mechanical error. Take a look at this reference chart showing exactly what hemagglutination looks like. A negative result remains a smooth, uniform red puddle. A positive reaction clumps together, looking like cracked pepper or distinct speckles in a clear fluid. The interpretation rules are straightforward. Clumping in the first drop indicates the presence of antigen A. Clumping in the second drop indicates antigen B. If both drops clump, you have type AB. If neither drops clumps, your blood is type O. The third drop works exactly the same way for your Rh factor. Visible clumping here means you are Rh positive. A smooth, unreacted drop means you are Rh negative. Here is a chart showing the full spectrum of possible ABO and Rh groups. Test yourself. Locate the slide showing type O positive. You'll notice there is no clumping in the A or B sections, but there is distinct hemagglutination in the anti-D section. Before we finish, always adhere to strict laboratory safety protocols. Dispose of all used lancets, toothpicks, cotton balls, and your glass slide in a designated biohazard container. You've safely executed a clinical laboratory procedure. By isolating your variables and correctly reading the physical reactions, you've successfully